So in this video we will be discussing about the notochordal tube and some general anomalies considered with the or related with the general growth of an embryo. So as I already said, some of the epiplastic cells which fall into the primitive pit, they will be moving cephalically till the precaudal plate, and that process or tube is called as notochordal tube or notochordal process or notochordal canal. Beyond the precaudal plate, they do not extend. So between the endoderm and the epiblast, this notochordal process extends cephalically from the primitive node to the precaudal plate. So once the notochordal tube extends up to the precaudal plate, it cannot extend further. Of course, there is a presence of a membrane after the precaudal plate, and it is called as buccopharyngeal membrane. Which will develop into future mouth. So see, I am just making a cross section. Why this notochordal process is very important? Because it will act as a central axis of the body around which will where the vertebra will develop, and on one side it is considered as the right side and on the other side it is considered as the left side of the body. So another important thing we must all know is mesoderm is present everywhere but except at three places. Mesoderm absent at three places. The first place is the precaudal plate. Where the endoderm and ectoderm are sticky together. At the very caudal area also there is a place where the ectoderm and endoderm are sticky together, and this place is called as cloacal plate, which will develop into anus in the future. So just more cephalic to the precaudal plate, it is the presence of the buccopharyngeal membrane. And there also the ectoderm and endoderm are sticky together and there is no mesoderm. So this membrane, buccopharyngeal membrane, will be developing into mouth in the future. Then another important place we must mention is where the notochord passes right in between the ectoderm and endoderm, where the notochord process, there also mesoderm is absent. So where notochord is present, there also mesoderm is absent. The ectoderm and endoderm are sticky together in that place also. So now I am going to make a cross section like this just for better understanding. So I hope everybody can understand the diagram. So this is the cross section, this is the ectoderm, this is the cloacal membrane, below there is no mesoderm, and this is the primitive streak and this is the primitive node. This is the ectoderm, this is the ectodermal layer, and this is the notochord extending from the primitive streak to that of the precaudal plate, and here it is the buccopharyngeal membrane once again. Buccopharyngeal membrane which will develop into mouth in the future, and this layer is the mesodermal layer. It is present apart from the four places, that is, it is not present under the cloacal membrane, where a place where the notochordal process is present, and a place where the precaudal plate is present, and a place where the buccopharyngeal membrane is present. It is not present over these four places, and this is the endodermal layer. So you all know, I hope, this is the amniotic cavity, and this is the yolk sac. So do you know, at the starting, the amniotic cavity will be connected to the notochordal process through the primitive node. So the amniotic cavity and the notochordal process will be connected through the primitive node. This is the notochordal process. So what happens as the time passes, the floor of the notochordal tube and the endoderm fuses together. They will fuse together like this. As the time passes, they will be fusing together, and both together they will disintegrate. Floor alone will be disintegrating. So now what happens? The amniotic cavity will be temporarily connected with the yolk sac. This happens only temporarily, and this is not a permanent connection. So the amniotic cavity will be communicating with the yolk sac. The fluid from the amniotic cavity, that is the amniotic fluid, will be flowing through the primitive node, then through the notochordal process, and then to the yolk sac. So the canal from which the amniotic cavity will be communicating with the yolk sac, that is the canal through which the amniotic fluid from the amniotic cavity will be flowing through the primitive node, the notochordal process, and then to the yolk sac. The canal name is, so this will be the canal, so connecting the amniotic cavity and the yolk sac. So the name of the canal is neuroenteric canal. Why is it named so? Because this is the side from where the future nervous system will be developing. In the future, the nervous system will be developing from this side. That is over the ectoderm, right? That is above the ectoderm. That due to the specialized secretions of the notochordal process, then neural tube will be developing, and that is a somewhat special function and differentiation of cells. And on this side, the GIT will be developing, that is the gastrointestinal system. That is from the endoderm that will be developing, right? So as this canal communicates from the part from where the nervous system will be developing in the future, it's communicating from the part where the nervous system will be developing in the future to the part where the future GIT will be developing. So this canal is called as neuroenteric canal. And this communication lasts only temporarily, and this is not permanent. This occurs for just some time, few times it occurs for some time. So I would like to show this process in 3 stages. Just to consider this as an ectoderm. And this is the mesoderm. So please remember those 4 places where the mesoderm will not be present. It is very much important. And this is the endoderm. Again and again I'm saying, just for my convenience I'm just scribbling the cells like this. So this is the ectoderm, this is the mesoderm, and this is the endoderm. 3 germ layers, and this is the notochordal process. So this is considered as stage 1, right? What happens during stage 2? Let us copy this. What happens during stage 2? This endodermal and the lower part of the notochordal tube will be fusing together, and they will be disintegrating, right? So the floor disintegrates. From this point of view, the floor disintegrates. So the notochordal tube will be exposed to the exterior. Why I'm showing these lines is, the neuroenteric canal is just passing through the notochordal tube. They will be just carrying the amniotic fluid from the amniotic cavity to the yolk sac. That's what I'm just drawing these lines, just to mention the amniotic fluid flow. This is considered as stage 2. Then what happens during stage 3? So this is the ectoderm. This is the mesoderm. And this is the endoderm. So what happens during stage 3? The notochordal tube or notochordal process just intercalates with the endoderm. So once the notochordal tube just starts intercalating with the endoderm, the neuroenteric canal starts disappearing. So this cell, once intercalation has been completed, this cell starts proliferating very rapidly. Of course, these cells will be replaced by the endodermal cells, but before that replacement, this proliferates very rapidly. So consider this as ectoderm. Then this is an endoderm, and this was the notochordal tube before, right? So it has been intercalated with the endoderm, and consider this as the mesoderm. This is the mesoderm. So what happens? This cell starts proliferating very rapidly. So this leads to the formation of a central solid rod-like structure. And now this rod-like structure is called as definitive notochord. Of course, it is a primitive structure. Notochord is a primitive structure which develops during the 3rd week of the intrauterine life. But why this is much important? Because it determines the axis of the embryo, central axis of the embryo. So usually it degenerates in the adults. All of us will be having a remnants of notochord. So that is called remnant of notochord. In adults is called as, it is called as nucleus pulposus. So nucleus pulposus, where it is present? It is present in the intervertebral disc. At the center there will be a jelly-like substance, right? So that is called the nucleus pulposus, which is the remnant of the notochord, which is usually seen in the adults. So then the definitive notochord, the adjacent mesoderm will be triggered to form the vertebral canal, and the ectoderm above will be triggered to form the CNS, that is the central nervous system. Of course, this definitive notochord will be triggering the ectodermal cells to form the notochordal plate, and that will be discussed in the development of nervous system. So after this, what happens? The embryo just starts folding by itself. So how that folds? I hope by this diagram you can just imagine how the embryo will be folding. This is the ectoderm. Okay. This is the endoderm, from which the gastrointestinal system, that is the enteric system, will be developing, and this is the mesoderm. Of course, there will be the presence of the notochord at the back. Okay, let me draw that also. So development of nervous system will be discussed in different video. So in this video, I'm just only discussing about the folding of the embryo, how the embryo folds. That's all I'm just discussing about that. So after this, what happens? So the embryo has folded fully. So this is the ectoderm. Of course, from skin is also a derivative of the ectoderm. So this is the endoderm. Of course, limb buds also start developing. The skin is derived from the ectoderm, and this is the mesoderm. Muscles, some of the ribs are also derived from the mesoderm. You know that the paraxial mesoderm, they give rise to somites, and the somites which just secrete the mesenchyme. That mesenchyme will give rise to different types of bones, and of course the development of bones and the neural crest cells also take part in the development of bones, and etc. That all will be discussed in some videos, in the next videos, in the forthcoming videos. But I'm just now discussing about the folding, how the folding happens, and how the limb buds are derived, and how the muscles are derived. I'm just discussing about that. Detailed lecture of the derivative, how the development of a neural system occurs, everything will be discussed in separate video. So this is the notochord. So as you all know, this is the definitive notochord, and it will be triggering the formation of the neural plate, and from which the central nervous system as a whole will be developing. That is the spinal cord and the brain. And this is the endoderm, from where the gastrointestinal system will be developing, and this is the mesoderm, from where the muscles and the urogenital system will be derived. So by the end of third week and the beginning of fourth week, and beginning of fourth week, by the end of third week and beginning of fourth week, gastrulation ends. As a revision, so let us revise what are all the steps that takes place during the gastrulation. So first is the formation of primitive streak and a node. Primitive streak and a primitive node. Then of course the cells within the primitive streak and node just starts disappearing, and they will be resulting into, primitive streak will be resulting into primitive groove. And the primitive node will be resulting into primitive pit. So then a formation of endoderm occurs by replacing the hypoplastic cells, and then formation of a mesoderm. The beautiful thing is that both are derived from the epiplast. Then of course once the mesoderm is formed, the remaining layers of the epiplast is called as ectoderm, and that's what ectoderm is also formed. Then formation of notochordal process or tube, which extends from the primitive pit or a primitive node to that of the precaudal plate. So these are all the things which happens during the process of gastrulation, and the anomalies will be discussed in the next video. Do subscribe and stay in touch. Thank you.